Hi students, coming to the next class is genetic code. In the last class we discussed about transcription process, right? In transcription process we discussed about the mRNA synthesis. There is an initiation, elongation and the termination process. And the next one we discussed about the post-transcriptional modifications process, because there is an 5' and cap is there and 3' and poly A type structure is present, right? And these two structures are involved in the stabilization of mRNA structure, right? It enhances the lifespan of mRNA. And next one is that there is a group of non-coding sequences are there. The non-coding sequences are called as introns, right? See, the introns removing process, that is nothing but splicing mechanism, right? A group of proteins are involved. That protein complex is called as Spliceosome Complex. See, that is about that coding and non-coding sequences of mRNA. So within mRNA there is a coding sequences are there. The coding sequences contains codons. The coding sequences contains codons. So now we are discussing about the genetic code. Now we are discussing about the genetic code, right? See, within genetic code, the term given by the genetic code term given by the George Gamow. George Gamow is the scientist name, and the genetic code discovery done by Nirenberg, right? Mathai and Corona. Okay, Hargobinda Corona, not Corona, right? Hargobinda Corona is the scientist name, right? So here Hargobinda Corona is one of the best scientists regarding to that artificial genes, right? He first did the artificial mRNA synthesis. Hargobinda Corona first did the artificial mRNA synthesis, right? So these three scientists done the research regarding to that genetic code, and the scientists are discovered the genetic code. Nirenberg, Mathai and Corona discovered the genetic code, right? That is the introduction for the genetic code. And coming to the types of amino acids. Types of amino acids. If we see in any cell, this is the nucleus. This is the nucleus, right? This is the genetic material. This is the genetic material, right? And that genetic material, okay, replicated. That genetic material replicated, that is called as DNA replication process. That genetic material involving transcription process, involved in the mRNA production, Okay, the mRNA passes the message from nucleus to cytoplasm. The message passed in the form of codons. So mRNA contains codons. See, mRNA contains codons, especially 4 nucleotides are there. Within RNA, 4 nucleotides are there. All we know, there is an uracil is there, right? There is an cytosine is there, and there is an adenine is there. There is an guanine is there. Uracil is there, cytosine is there, adenine is there, guanine is there. See, the basic differentiation between DNA and RNA. Within DNA, thymine is present, uracil is absent. Within RNA, uracil is present, thymine is absent, right? So here uracil, cytosine, adenine, guanine, these 4 are present, right? So this is the nucleus. Nucleus contains the genetic material, that is the DNA, and DNA it's involved in the transcription process. Okay, it produces the mRNA. mRNA contains the message in the form of codons. That mRNA enter into the cytoplasm, involved in the protein synthesis process. Means that mRNA contain codes for amino acids. These amino acids are involved in the protein synthesis. The amino acids are joined by the ribosomes, contains peptidyl transferase enzyme, right? So that is nothing but translation process. So here concept is, in a cell 20 types of amino acids are coded by codons. In a cell 20 types of amino acids are there which are coded by the codons. All we know according to the NCRT, Okay, 20 types of amino acids are present within that cytoplasm, right? These 20 amino acids are coded by the codons. These 20 amino acids are coded by the codons, right? So that is about that 20 types of amino acids coded by the codons. And see how the coding process is done. Generally, we already discussed four nucleotides are not sufficient to code 20 amino acids. Here only 4 nucleotides are there. Okay, directly these 4 nucleotides unable to code for 20 amino acids. Directly these 4 nucleotides unable to code the 20 amino acids directly, right? So here the very first case is 4 into 1, right? See 4. So in that case the 4 nucleotides are unable to code for the 20 amino acids. And next case there is in 4 square, here 4 into 4 16, right? These 16, the 16 codons also unable to codes for the 20 amino acids, because here 16 codons are there, 20 amino acids are there. So the 16 codons are not sufficient to code. The 16 codons are not sufficient to code 20 amino acids, right? And next one 4 cube. And next one is the 4 cube. So in that case 4 into 4 total 64 codons are there. Total 64 codons are there. The 64 codons are codes for 20 amino acids. The 64 codons are codes for 20 amino acids. So what I am telling here, whatever that genetic code is there, the genetic code here indicated, okay, triplet. Always genetic code is triplet. Always genetic code is triplet. For suppose here U, C, A, G, these are individual. Okay, if these are individual, okay, these are not sufficient to codes for 20 amino acids. In the second case, maybe the U, C, or U, A, or U, G, like this. Totally here 16 types of codons are found in the second case. The 16 types of codons are not sufficient to code 20 amino acids. Not sufficient to code 20 amino acids, right? So here the third case 4 into 4, totally here the genetic code is the triplet. Genetic code is the triplet, 4 cube, right? There is a genetic code AUG or UUU. So if the genetic code is triplet, totally 64 codons. Totally 64 codons are formed, right? Genetic code is doublet. That case, okay, not possible to code all 20 amino acids. Genetic code is triplet. In that case, okay, codes for all 20 amino acids. Genetic code codes for all 20 amino acids. So that is the very first point. That's the reason why always genetic code is triplet nature. Always genetic code is triplet nature, right? That is the very first point. And coming to the next point is genetic code is universal. Genetic code is universal. Universal means we know different types of organisms are there. Prokaryotic cells are there, eukaryotic cells are there. In eukaryotes, again different types of organisms are there. Okay, in any organism, these codes are very specifically codes for specific amino acids. Either in prokaryotic cells, either in eukaryotic cells, the codes are same. The codons are same. Okay, we are thinking the prokaryotes are different from eukaryotes. Definitely, prokaryotes are different from eukaryotes, but not regarding to the genetic code. Genetic code is same for all living organisms. Genetic code is same for all living organisms, right? So that's the reason why here genetic code is universal. Genetic code is universal. And next one is non-ambiguous. We know about the ambiguous. Ambiguous indicates here confusion. Ambiguous, the person is ambiguous state. Means here confusion state. See, non-ambiguous, without any confusion. Without any confusion means very specific. Here genetic code is very specifically codes for specific amino acids. Genetic code is means each codon is codes for, okay, each codon specifically codes for specific amino acids. So here non-ambiguous. Here non-ambiguous indicates here very specific. Here very specific. specifically codes for specific amino acids, specifically codes for specific amino acids, right? So that is about the non-ambiguous, right? And non-overlapping. Here, non-overlapping indicates, one of the best example is, this is the 5' end, this is the 5' end, right? So in all the cases, every mRNA contains the very first codon is the AUG, very first codon is the AUG, and next one, this is the initiation codon, either in prokaryotic cells, either in eukaryotic cells, right? The initiation codon is the AUG, right? And next one, maybe the codons are changed from one type of mRNA to the another type of mRNA. UU is there, UCA is there, UGA is there, right? And maybe AAA is there, right? Like this, AAA and 3'. So this is the one of the structure of mRNA. mRNA contains the codons. mRNA contains the codons. This is the one codon. Yeah, this is one codon. This is one codon, and this is another codon. This is, yeah, these are the different codons. These are the different codons. Maybe here not G. Okay, if G is there, that one is maybe different. So here this one, A, UAA, not A. Yeah, C, UCA, right? Here taken, okay, means UAA, UAG, UG, all these are stop codons. That's the reason why, right? I mentioned here C. Okay, I'll explain what is stop codon, right? See, AUG, UUU, UCA, yeah, UCA, UCA, AAA. Maybe the same codons also repeated sometimes, right? So here AUG, UUU, UCA, UCA, and AAA. That is the one of the mRNA sequence. So always genetic code is non-overlapping. Always genetic code is non-overlapping. Here overlapping means, see, these three codons are not involved in coding of amino acids. Again, these three are not overlapping. Not overlapping. So see what I am telling here. Always genetic code is very specific and non-overlapping. The three codons are codes for one amino acid. So the genetic code, okay, or the codons are not depends on neighboring codons. Not depends on neighboring codons. The three codons are specifically codes for specific amino acids. Specific amino acids. There is no chance for the overlapping. There is no chance for the overlapping. So here there is no overlapping process. There is no overlapping process. So the triplet codons, yeah, triplet codon. This is one of the three nucleotides are there. This is the triplet, right? So genetic code is always triplet, and genetic code is universal. Genetic code is universal, right? And genetic code is very specific. Genetic code is very specific, and genetic code is non-overlapping. Genetic code is non-overlapping, right? And genetic code is commaless. See, between these two codons there is no comma. Between these two codons, like this. Between the codons there is no comma. There is no full stop. So no punctuations between the genetic code or between the codons. Between the codons, okay, there is no punctuations. Commaless. Code is commaless, right? And code is degenerated. Code is degenerated, or code is redundance. Code is degenerated or code is redundance. What is degenerated or what is redundance? See, generally we know about initial 20 amino acids are there. Here total in this table 64 codons are present. Total in this table 64 codons are present, right? So in that 64 codons, 3 codons are nonsense codons. 3 codons are nonsense codons. 3 codons are nonsense codons. See, nonsense codons means which are not codes for amino acids. Which are not codes for amino acids. Which are involved in the termination process. Means whatever the translation process is there, the translation process stopped by the 3 codons. See, this is the first stop codon, this is the second stop codon, and this is the third stop codon. So in the entire genetic code, 3 codons are Three codons are nonsense codons, which are not codes for amino acids. So these three codons called as nonsense codons, which are involved in the termination of translation process. These are also called stop codons. So here total codons are 64. 3 are nonsense. Remaining 61 codons are there. 61 codons are sense codons. 61 codons are sense codons. Here these 61 codons are codes per 20 amino acids. 61 codons are codes per 20 amino acids. So here one amino acid is coded by more than one codon, because 61 codons codes per 20 amino acids. 20 amino acids means here more than, each amino acid, each amino acid coded by more than one codon. Each amino acid is coded by more than one codon, because here 61 codons are there, 20 amino acids are there. So each amino acid, coded by more than one codon, right? So that is about the code is degenerate or code is redundant. Code is degenerate or code is redundant. Very simple. Each amino acid coded by more than one codon. Right? So that is about the some of the important points which are related to the genetic code. Some of the important points which are related to the genetic code or characteristic features of the genetic code. So here, coming to the next one, we need to explain the entire genetic code table. This is the table. So here, especially how many number of nucleotides are present in RNA? 4. How many number of nucleotides are present in RNA? 4, right? So here, see, I did not mention initially here because I don't have space. That's why I mentioned later. I mentioned now here. UCAG, right? UCAG. This is about that genetic. This is the entire genetic code, right? So how the genetic code, how genetic code forms the codons? We know. See, in the entire RNA structure, 4 nucleotides are there. These are UCAG. See, always genetic code is triplet. The very first one, U. Right here, U, and C, and A, G. See, always here blue color line indicates for the first nucleotide. Always the blue color line indicates the first nucleotide. And next one, U, C, A, G. Right? So here, UCAG indicates here, that is about the second nucleotide. See, in every case, the black color letter, that one indicates the second nucleotide. See the last one, UCAG. Each table, this is the one box, UCAG. This is the one box, UCAG. So this is the one box, right? UCAG. So the red color letters are indicates the third nucleotide, right? So see, by this way, the total codons are formed. So in that case, the very first one is the U. This is the first letter U. U, second letter U, right? The first letter is the U. Here first letter. Here last letter. This is the first letter, right? Here last letter. In genetic code, in codon, this is the last letter. U, right? C, right? U, right? A, right? U, G. So here, by this way, the total codons are formed. Here U and UUC, this one forms the phenylalanine. Right? And UUA and UUG, right? C, here this one is the C, U, C, yeah, CUC, right? CUA and CUG, right? CUU, CUC, right? CUA, right? CUG, right? So here CUU, CUC, CUA, CUG, and these are these are the first two, right? Totally 6 codons or codes per leucine amino acids. 6 codons or codes for the leucine amino acid. We discussed genetic code is redundance. Genetic code is redundance, right? So here genetic code is degenerate or genetic code is redundance means, okay, one amino acid, here one amino acid, the amino acid name is leucine. The amino acid name is leucine. That leucine amino acid coded by 6 codons. The single amino acid coded by the 6 codons. So based on this one, we can say genetic code is redundance. Genetic code is redundance. Or genetic code is degenerate. Genetic code is degenerate, right? So that is about that. The leucine, here 6 codons are there. And next one, this is A, U, again U, right? A, U, C, A, U, A, and A, U, G. See, this is the case is different. See, A, U, U, A, U, C, and A, U, A. These 3 are codes for isoleucine. These 3 are codes for isoleucine, right? The AUZ, the codon is quite different from the remaining codons. AUZ is also called initiation codon. Initiation codon. AUZ is the initiation codon. Why this is called as initiation codon? During transcription process, during translation process, the very first coded amino acid is the very first coded amino acid or very first coded codon is the AUZ. AUZ is codes for methionine. AUZ is codes for the methionine. AUZ is codes for the methionine. So what I am telling here, whatever that prokaryotic cells are there, whatever that eukaryotic cells are there, either in prokaryotic cells, either in eukaryotic cells, the translation takes place. During translation process, the first coded codon is AUG, right?